Very good morning to all. Today the topic of discussion is on cancer, and we will be looking in detail the 6 hallmarks of cancer in the coming slides. Now, what is cancer? So throughout our life, healthy cells in our body divide and they replace themselves in a controlled fashion. Cancer starts when a cell is somehow altered so that it multiplies out of control. Now a tumor is a mass composed of a cluster of such abnormal cells. Now most cancers form tumors, but not all tumors are cancerous. So they are either benign, meaning they are non-cancerous. So they don't spread to other part of the body and they do not create new tumors. But in case of malignant cancer, these tumors crowd out healthy cells, they interfere with body function, they draw nutrients and they can spread to any distant organs. So such process of invasion of cells to different part of the body is called as metastasis. There are different major types of cancer such as carcinoma, sarcoma, melanoma, lymphoma, leukemia, etc. Now carcinoma is the most commonly diagnosed cancers. So they originate from the epithelial cell lining of the internal surface of various organs such as skin, lungs, pancreas and other organs and glands. Meanwhile, sarcoma, they arise from the mesodermal cells constituting the various connective tissue such as the fibrous tissue, bone, muscle, fat, blood vessel, cartilage and other soft connective tissues of the body. Leukemia, lymphoma and myeloma, they arise from cells of bone marrow, lymphatic system and the immune system of the body. And melanoma, they are the cancers specifically for skin. What are the causes of cancer? Now cancer is caused by accumulated damages to the gene. So such changes may be due to chance or to exposure to cancer causing substances. Such substances that cause cancer, we call them as carcinogen. So a carcinogen may be a chemical substance such as certain molecules in the tobacco smoke. They can also be because of other biological factors, internal factors, especially in terms of age, gender, inherited genetic defects and even the skin type when it comes to skin cancer and such things. It can also be due to environmental exposure, for instance to UV radiation and fine particulate matters. There is also the lifestyle related factors such as your food habits, smoking, your drinking habits, use of tobacco and such things. There are other factors that also will lead to this cancer. Say for example, the bacteria and virus can also cause cancer. Heliobacter pylori, which can cause gastritis. The hepatitis virus, the human papilloma virus, which cause cervical cancer, and so on. So cancer can be because of lot of different factors. They can also be due to radiation, which we have already discussed. The UV radiation can cause cancer, even the X-ray radiation can cause cancer, even the non-ionized radiation from the sun can cause cancer. Even taking some drugs may also increase the chance of cancer. Certain hormone treatment or medicine that cause immune deficiency, all those can be a reason for causing cancer. So when you learn about cancer, it is very important that you know about what are oncogenes and what are tumor suppressor genes. Now proto-oncogenes are like if a normal gene which is altered by mutation, they can become a oncogene and that can contribute to cancer. So proto-oncogene may have different function in a cell. Say for example, some proto-oncogenes provide signal that lead to cell division. The other proto-oncogene can even be involved in different cell death pathways, specifically programmed cell death pathways such as apoptosis. Now the defective version of these proto-oncogenes are what we call as oncogenes, and they can cause a cell to divide in an unregulated manner. This growth can occur in the absence of normal growth signal such as those provided by growth factors. The key feature of oncogene activity is that a single altered copy leads to unregulated. Now when it comes to tumor suppressor genes, these are normal genes that slow down cell division, they repair DNA mistakes, or they tell the cell when to die, such as a programmed cell death pathway where the cell can die due to apoptosis, a cell can die due to necrosis or paraptosis, and so on. Now, when a tumor suppressor gene don't work properly, cells can grow out of control, which in turn can lead to cancer. A tumor suppressor gene is like the brake pedal on a car, and we can say the oncogenes or the proto-oncogenes are like the petrol in the car, they or the acceleration pedal in the car, which helps to accelerate the growth. Now here it normally keeps the cells from dividing too quickly, just as the brake keeps a car from going too fast. When something goes wrong within the gene, such as a mutation, cell division can get out of control. An important difference between oncogene and tumor suppressor gene is that oncogene result from activation, which is in fact turning on off proto-oncogenes, but tumor suppressor genes cause cancer when they are inactivated or when they are turned off. So our human system maintains a homeostasis or maintain a healthy condition if they can maintain the proto-oncogene level as the tumor suppressor gene level in what is required. In case if the proto-oncogene activation to oncogenes takes place to beyond a limit, then it leads to uncontrolled proliferation, or even in case the suppressor gene deletion happens, it also lead to uncontrolled proliferation. Now for example, as we have seen in the last slide, p53 is a tumor suppressor gene, and if there is an abnormality in this p53 protein or the TP53 gene, it can lead to cancer, and almost half of all the cancers that is known have their abnormality in the p53 genes. Now this is a pictorial representation of the involvement of both tumor suppressor as well as proto-oncogene. So in a normal cells or in a normal cell cycle, the proto-oncogene and the tumor suppressor gene, they complement well so that we have a proper regulated growth. Now in terms of cancer, the mutation can lead to cancer. So any mutation, say for example the proto-oncogene, if it turns to oncogene, too much of gas, meaning too much of acceleration, and it can be dangerous to the cell. And if the brakes go off, the tumor suppressor genes are inactivated, again there is uncontrolled proliferation of cell, which can lead to. Douglas and Weinberg are two prominent scientists where they published the Hallmarks of Cancer in a journal Cell in early 2000. So they believed that the complexity of cancer can be explained in a small number of underlying principles, which we call as hallmarks of cancer. Now these are the first 6 hallmarks of cancer that they proposed, which means cancer can sustain proliferative signaling, they can evade growth suppressors, they can activate invasion as well as metastasis, they have replicative immortality, they induce angiogenesis, and they resist cell death. The first hallmark of cancer is sustaining proliferative signal. So the growth signaling in normal cells is a highly regulated process wherein proliferative signals are activated whenever necessary and deactivated when no longer necessary. So this tight regulation ensures cell homeostasis. However, in case of cancer, this regulation is compromised. So one of the fundamental trait of cancer cell is their ability to proliferate without a controlled signaling input. Now they achieve this in a number of ways. So they can achieve this by increasing growth factor production, by stimulating the normal cells in the microenvironment to provide them with growth factors. So the increasing the number of receptors on the cell surface so that they can bind to more of the ligand, and structurally altering the receptors to facilitate cancer cell signaling. They can also activate proteins in the downstream signaling pathway so that they can sustain these proliferative signals. So one of the most prominent of these signaling channels is the growth promoting signal transmitted through the RAS-RAF-MEK pathway, and recent studies also highlight the ability of cancer cell to disrupt the negative feedback loop that act as a safety mechanism to downregulate the signaling pathway whenever a mitogenic signal is hyperactivated. So one key example of this is the RAS oncoprotein, where there is a disruption in the normal negative feedback mechanism operated by the oncogenic GTPase. So this slide give you the pictorial representation of what we have discussed in the earlier slides. So on top left we can see the growth factors. The EGF here binds to the EGFR receptors, and then that they result in the cascade of reaction including the RAS, RAF, MEK, ERK, and ultimately the cell growth and proliferation. There is simultaneously another pathway, the PI3K, KT, mTOR pathway, that also result in cell growth and proliferation. So in case of any mutation in the EGFR receptors or RAS or PI3K, or in case of, as we have seen or as we can see in the down picture, this is what we have discussed in the earlier slide. There is always a negative feedback loop which makes sure that in normal cells their pathway does not lead to high proliferation of cells. In terms of cancer, the negative feedback loop is missing, which in turn lead to high proliferation of cells. So it is all about how the cells or how the key molecules in the signaling pathway are mutated so that it becomes beneficial for the cancer cells. Now the second hallmarks of cancer that we'll be discussing is evading growth suppressors. Now cell proliferation in normal cells is a tightly controlled process wherein the pro as well as the anti-inflammatory signals are coordinated properly, and their activities at the cell cycle level are strictly under control. Now there are two tumor suppressors most commonly dysregulated in cancer cell, which are the retinoblastoma protein, the RB, and the tumor protein, the TP53. The RB actively inhibits cell passage through the restriction point in the G1 cell cycle phase, and once the RB is mutated, it removes this gatekeeping activity and it allows the cells to undergo the uncontrolled proliferation. So which means if there is any defect in the RB pathway, the cell cycle progression will be persistent and the cell keeps on proliferating. Now when it comes to T53, the T53 protein function as a cell regulator of apoptosis because it arrests the cell cycle in the cells when there is a DNA damage. And if there is a loss of this P53 tumor suppressor gene, either through mutation or some other factors, it allows for cell cycle progression despite the DNA damage or cellular stress that is detected in our body. P53, as you all know, is a transcriptional factor that is activated by diverse genotoxic as well as cytotoxic stress. Now upon activation, the P53 prevents the proliferation of these cells. They initiate the cell cycle arrest, they initiate apoptosis, as well as they may also initiate DNA repair. Now this P53 also will help the MDM2 gene to promote its transcription. Now these there is an antagonistic action of MDM2 towards P53, which we can call as a negative feedback loop, is set up between MDM2 and P53. So imagine like if there is a P53 that activates MDM2, this MDM2 which in again target P53 for activation. So P53 helps the MDM2, but MDM2 target P53 for degradation, which means there is always a negative feedback loop which regulate how much of P53 should be active and so on. So this feedback loop is likely the key mechanism for controlling or restraining the P53 activity in normal cells in the absence of the earlier discussed stress like DNA damage or cell cycle abnormalities or even like hypoxic conditions. Now when it comes to the RB regulation of cells, these are again like a negative regulatory protein that control the cell division, and they can also be considered as a tumor suppressor protein. So RB is active when it is in a dephosphorylated state, and when it is in a dephosphorylated state, it binds to E2F, which is a transcriptional factor required for the transcription and eventual translation of molecules that play a key role in the cell cycle. Now E2F cannot transcribe certain genes when it is bound to RB. So as you can see in the figure, E2F when it is bound to RB, it does not activate the proteins that are required for the cell cycle progression. So and the unphosphorylated RB binds to the transcriptional factor E2F, and meanwhile if the RB is phosphorylated, and if it becomes phosphorylated, the cell growth triggers the phosphorylation of RB. In fact, cell growth in the sense if the cell grow, then it has to divide, and our system detect, okay, now the cell has like grown in size now, and since it has to go to the cell cycle or cell division, the RB becomes phosphorylated and then it is inactivated. The inactivation lead to the release of E2F, and they can promote the transcription of gene, its control, which are involved in the cell cycle. The third hallmarks of cancer we will be discussing is resisting cell death or evading cell death. Now normal cells may initiate apoptosis in response to DNA damage and other cellular stress, while if you look at cancer cell, they are generally less sensitive to this sort of stress, and they tend to avoid the programmed cell death pathway such as apoptosis, necrosis, autophagy and so on. Now apoptosis occurs through two pathways, specifically we call as intrinsic or the mitochondrial pathway. So these are initiated by intracellular stress and other factors which may be quite important when it comes to cancer, and the extrinsic pathway where the death receptor pathway that is initiated by cell surface receptors which bind to specific ligands. Now there are a lot of pro-apoptotic proteins like BAX and BAC, so they disrupt the mitochondrial membrane integrity, which leads to mitochondrial membrane depolarization, which ultimately lead to caspase activation through the release of cytochrome C or other apoptosis inducing factors. Now as I already said, the DNA damage and the growth factor deprivation, chemo and as well as immunotherapeutics are activators of the intrinsic pathway, and these intrinsic pathways is tightly regulated by a group of related proteins which we call as BCL2 family proteins. Now cancer cells are taught to resist these apoptotic programmed cell death pathway mainly through two mechanisms. So either by the downregulation of the pro-apoptotic protein, or they can increase the pro-inflammatory signals or the surviving signals. Say for example, if you have a mutated P53, it can result in cells that are less sensitive to the DNA damage, and also they downregulate even caspase 3, which also ultimately inhibit apoptosis. So these are the different mechanisms in which the cancer cell thrive. They evade the normal cell death path. This is a pathway that shows both the intrinsic as well as the extrinsic apoptotic pathway. You just have to have an overall idea of like how the apoptosis happens. Now, if you look at the extrinsic pathway, the signals from the surrounding environment of the cell will bind to a death receptor, and that's why we call it as an extrinsic sort of pathway on the surface of the cell membrane. And this causes the conversion of the inactive procaspase 8 into a active caspase 8. Then this caspase 8 goes on to activate the caspase 3, which is effector caspase, which begins the caspase cascade that lead to the cell death or apoptosis. In intrinsic pathway also there are caspase involved, but then the caspase involved initially are different from what we have seen in the extrinsic or extrinsic apoptotic pathway. In the intrinsic pathway, typically it's initiated by internal stress such as DNA damage or reactive species generated inside the body. The p53 is activated, which activates the pro-apoptotic protein bags, which can initiate the release of cytochrome C from the mitochondrial membrane, inside of the mitochondrial membrane, because it can result in change in mitochondrial membrane potential, and it can lead to leakage of these proteins such as cytochrome C or apoptotic inducing factor that are embedded in the inside the membrane of mitochondria. Now, APAF1 and the release cytochrome C combine to form a complex which we call as apoptosome, which helps in the conversion of the inactive procaspase 9 into an active caspase 9, and this active caspase 9 activates caspase 3 in this extrinsic pathway. Unlike there is no role of caspase 9 in the extrinsic pathway, so the caspase involved in the activation of caspase 3 in these two cases are different. So but after that, the caspase 3, similar as in case of extrinsic pathway, lead to these cascade of caspases resulting in apoptosis. The fourth hallmark of cancer that we'll be discussing is enabling replicative immortality. Now, the normal cells have a finite replicative ability, meaning a intrinsic cellular mechanism allows the normal cells to divide a particular number of times or a finite number of times, and then they will block the cell division after reaching a limit. But in case of cancer cell, they overcome the sort of limitation or the sort of limit by overexpressing telomerase, an enzyme that maintains telomere length, which protect the end of the chromosome, and that will allow the cell to continue proliferating. And this process is also aided in part because of the loss of function of the tumor suppressor gene, which is the TP53. There are also some of the additional function that has been described recently, where the telomerase will enhance the cell proliferation also, and also they will help to resist apoptosis. And it makes sense because there is always, or there is a P53 mutation that is happening when there is a replicative immortality, and that's how it can help the cell proliferation, or it can resist apoptosis. And also they are also involved in DNA repair mechanism. As you can see in this figure, compared to the normal cells, the cancer cell, even after there is a damage, the telomerase is active, and which can give them an immortality. Usually after a particular finite number of replication, the cells either go to senescence, or they are going to die through apoptosis. But in case of cancer, such pathway or T53 is inactivated, and hence the telomerase will lead to immortality of cells, and which is a characteristic of cancer. The fifth hallmarks of cancer that we'll be discussing is inducing angiogenesis. Now, angiogenesis is the extension of the existing blood capillaries. So, in case of tumor cells, the process of angiogenesis or this formation of new blood vessels is very critical because it helps for the sustained tumor growth as well as metastasis. Now, tumor angiogenesis is a multi-step process which involves signaling input from several what we call as pro-angiogenic growth factors, and some of those are the vasculoendothelial growth factor, which is VEGF, which helps in the angiogenic switch. Now, why do you need the angiogenesis, or why do you need new blood vessels? So it helps in the delivery of oxygen and nutrients through blood, and also it also helps in the production of growth factors that will benefit the tumor cells. Now, there are a lot of studies that has been carried out which have come to the conclusion that there are a lot of other roles that the neovascular system or the neovasculation is to play in case of cancer. So mainly there are two additional components that helps in this angiogenesis or neovascular formation. The one is pericyte, which are the surrounding cells that can have the long associated uh, uh, role when it comes to even normal tissue vascular system formation. However, this also helps in tumor angiogenesis. There is also an indication where the bone marrow derived cells such as the neutrophils and macrophages, they are also recruited to the site and also help in the so-called angiogenic switch. It also helps in it also helps in the production of interleukin 6, a metastatic stimulator, and also protects new cells from apoptosis, where the integrin signaling have a huge role to play. Now, matrix metalloprotease, a topic that we will be discussing when we learn about the extracellular matrix in detail. They helps in the breakdown of a basement membrane, which allows the migration of endothelial cells. They also help in the extension of capillaries, new blood vessel formation, since the MMPs through the breakage or cleavage of basement membrane release lot of growth factors that are involved in all these processes. There are also a lot of angiogenesis inhibitors, specifically a lot of statin and also the VEGF receptor kinase inhibitors and the MMP inhibitors that are already known or already in the. Now, the sixth hallmarks of cancer that we'll be discussing is tissue invasion and metastasis, and we have gone through an entire lecture on metastasis, so we won't be discussing in detail, but then this is one property or one characteristic that makes the difference between a benign and a malignant tumor. So there are a set of controls to ensure that these cells stay where they are supposed to stay in the body. So in case of benign, the tumor cells grows only locally and they cannot spread, they cannot evade, or they cannot metastasize. But in case of malignant cancer, the cells evade and invade the neighboring tissues, enters the circulatory system, and then they have their own colony established in a different site. Here again, MMPs of the cancer cell degrade the basement membrane, the thin layer of extracellular matrix, which act as the barrier for cell movement, and they help the tumor cells to enter the bloodstream, the circulatory system, and the circulating tumor cells get trapped in a network of capillaries and set up their own secondary tumor after they extravasate at a particular site. And there are other factors or other players that are involved. Say for example, the tumor associated macrophages, the TAMs, they also supply these cancer cell with epidermal growth factor and colony stimulating growth factor, which also helps in the intravasation. There are other key factors which we all have already discussed that play the major role in cancer or tumor metastasis. This again is a pictorial representation of how the metastasis happen. So if there is a primary tumor that is already formed, then this malignant tumor, they have a local invasion, again that with the help of macrophages and the cold stimulating factor, as we have already discussed, and the EGF will help in intravasation of these tumor cells. They intravasate into the circulatory system and they survive there, and there will be a lot of other factors that will they have to undergo so that they survive in the circulatory system. So once they survive in the circulatory system, they reach and are at a very distant organ site, and it all depends on the seed to soil theory, where actually the secondary tumors are formed. But then after they are at a distant organ site, they extravasate from this capillary system, and then there is a micrometastatic formation, which also is aided by the matrix metalloprotease, and they form a colony in those area where they have localized, and again they develop into a secondary detectable tumor. So in conclusion, certain form of cancer may be less dependent on one hallmark or the other, but then some of the hallmarks do support how a particular cancer function. And the hallmark concept is helping to rationalize the new mechanical data from the cancer research community. And we also expect a lot of revisit to hallmarks of cancer. There is a chance that more and more hallmarks of cancer can be added depending on the new discoveries and new exploration that happens in the field of cancer. Thank you.